Hello and welcome to Odds and Ends, Part 15. In this episode, we'll cover the domestication of Noni, Feijoa, and Jamoon. We'll start with Noni. Noni is part of the genus Morinda, specifically Morinda certifolia. It's general knowledge that Noni is a fruit that tastes terrible, but is highly nutritious, and as such has been used historically for starvation food as a last resort, and medicine. The researcher Anura K. Singh has studied, in a paper I'm referencing right now, the genetic diversity of Noni and its center of origin and domestication. And here are the results of his study. Based on the predominance of a high level of Morinda species residing in India, with India the second highest center of diversity of species of Morinda, second only to China, it is very probable that Noni was domesticated there. The wild form of Noni is also found native in the southeastern and western Ghat region in India. It was also found that within the Indian subcontinent, three forms of Noni were found: the wild form and the domesticated form, as well as all of the intermediaries and genetic approximations of intermediaries, i.e. hybrids between wild and domesticated forms. All the species of Morinda within India, including Morinda citrifolia, are morphologically and genetically very similar, further indicating that this is where Noni was domesticated. There are also writings and diagrams dating back to 1500 BC, showing an ancient use of Noni far longer than most of the other Asian locations. There's also evidence of trade between India and China under the barter trade, a practice that's still going on with Myanmar. Based on the current information, Southeast Asia seems to be the primary center of origin and domestication for Noni. Now on to the domestication of the Feijoa. The Feijoa is in the genus Aca and is of the species Aca silowiana. It is a species native to the South Americas, predominantly in Uruguay and Brazil, as well as parts of Argentina and Paraguay. Known for its sweet acidic flavor that's mostly consumed raw, but several commercial products contain Feijoa. It is commercially planted right now in China and New Zealand, although this crop is only partially domesticated. Dr. Joel Donalazzo sought to identify the genetic diversity of both domesticated and wild populations to see how much genetic variation there is in both groups and how to preserve the wild populations for further breeding purposes. And here are the results. In terms of genetic variation within populations, it was found that there was higher inbreeding between individuals within the managed populations versus the wild population. Inbreeding in this sense meaning crossbreeding between related trees. Also within the wild populations, there seems to be at least two distinct genetic groups within the wild populations, in comparison with one in the domesticated population. In terms of between populations, there was found to be a great amount of crossbreeding between the managed populations, which are divided into populations grown by farmers, populations grown in backyards, plants generated from mass selection, which have all continuously crossbred with each other. There also appears to be some crossbreeding between the wild and domesticated populations as well. The Chilean domesticated population, grown by farmers primarily, is the most differentiated population of Feijoa in terms of genetic distinctiveness from the original wild population. Within current genetic analysis, it seems that the Chilean domesticated form is derived from Uruguayan specimens of Feijoa. Now on to its breeding history. After the first European who discovered it, Mr. de Watt from Switzerland, collected and planted it in one of his greenhouses in 1887. In 1890, the at that time renowned horticulturist, Dr. Edward Andre, bought a specimen from La Plata, Brazil, and grew it in the Riviera in his garden. After it flowered and fruited, he wrote a description of the entire plant and recommended that everybody grew it. Later on, a nurseryman in Lyons named Benson Freres obtained cuttings and layer seeds from Dr. Andre and Montevideo, respectively, and raised thousands of plants. Later on, seedlings from Dr. Andre's plants were obtained by Dr. Francisci in Santa Barbara, California. It is from these populations that the domesticated form of was bred. Now on to Jamoon. The jamoon is a species of Syzygium called Syzygium cumani. It is native to the Indo-Malaysian region, where it's distributed pretty much everywhere within that region. It's a fruit common amongst these groups of people. As such, the researcher Dr. Safir Uddin of the University of Agriculture in Faisalabad, Pakistan, I hope I'm pronouncing that correctly, to find out genetic diversity, and here are the results. Based on the 50 strains from 10 major jamoon growing districts in Punjab, it was found that indeed it was first domesticated in Pakistan, with the area now known as Pakistan being the location of the greatest level of genetic diversity. It was found that the strains labeled SFM08, TUC01, SFG07, KKF02, MWF01 are the most predominant strains that have the best features for future breeding projects. The strains KDC03, TUC02, and TUC03 seem to be strains that are bred for small seeds. It was also found that genetic diversity within the domesticated populations is very small, consisting of only 3 distinct genetic populations. As such, it's estimated that future crossbreeding attempts would be between different domesticated groups to improve heterosis, or between the wild forms and domesticated forms. And that about covers everything. Thank you for watching. Thank you for watching this video. If you're watching on YouTube, please subscribe to me on BitChute for a greater variety of content, 4 videos a week. And thank you to all my subscribers on both platforms. I appreciate it.